So I was talking about genomic imprinting. Okay, there are several genes that are expressed from both the alleles. Some of them are repressed from both the alleles, as it depends on the developmental stage, cell type, and many other things. And some are expressed only from the paternal allele and repressed. The allele that is present on the maternal allele is repressed. Some are expressed only from the paternal allele and the maternal allele and paternal allele is repressed. So the methylation, usually it is about DNA methylation, and DNA is methylated on the cytosins, which are usually having CG, and several of those. In that, those are called as CpG islands, and those the cytosins are methylated. The transcriptional factors, general transcriptional factors, and many other proteins that are involved in recognition and transcription of a gene are usually specific to DNA, right? They do not recognize if they are. What I mean here is unmethylated DNA. Methylation is not a common thing that is present all the time, but in higher eukaryotes, and there are many other regulatory processes where methylation is involved. So if the DNA is unmethylated and there are activators and every other proteins, then transcription will go ahead. But if this DNA is methylated, then it will interfere with the binding of these transcriptional factors to the promoter elements. Therefore, repression would occur. So on a simplistic note, what we will think of is we will say that the predominant mechanisms of genomic imprinting is DNA methylation, because that is what we are concerned with. So I would want you to also think about it. And please note down bisulfite sequencing. If you have learned it, good. If you did not, please try to know about it. Self-study question, bisulfite sequencing. It's a simple one. So I hope you understand that if there is methylation, there is no expression. If there is no methylation, then we have expression, right? So when I say a sentence like gene A is paternally imprinted, what I mean by that is gene A on the paternal, this is the paternal allele of gene A. Paternal because it's coming from the father. Is imprinted, I said, which would mean that there is methylation in this region. If there is methylation, then we will not have any transcription. So I said gene A is imprinted, and I'm saying it is paternally imprinted, which would mean that there is repression in the paternal allele. So there is expression of gene A here from the maternal allele. Okay. So this DNA here, the promoter is unmethylated. Say, for example, if I said gene B is maternally imprinted. If I said, which would mean that there are two alleles, right? Paternal allele B and maternal allele B. So I said maternal allele is imprinted. So that would have been imprinted, and because that is methylated, because of which there is no expression of B. And from the paternal allele, there is no methylation, so transcription could occur, and expression of gene B from the paternal allele is there. Okay. So these are some questions that you don't have to immediately understand. These are some concepts. It will take a little bit time. I would want you to watch the videos that I will also post, so they should help in understanding. So what we need further is, let me see if I understood this, if we all are on the same page. I will have a small modification to this, and you can answer the questions that I might pose. Which gene is expressed? Which allele is expressing gene A? S. Gene A is expressed from the maternal
Maternal
allele.
one.
That's right. It is imprinted on the paternal allele. Gene B, on the other hand, is expressed from the paternal allele. Right? I hope that is clear. So we will also see a mechanism that happens, and I'm trying to include complexity so that we will try to solve a complex problem, and it'll be nice to do that. So the first thing that I would want to tell is, say there is an individual. This individual is biparental. That means that one is set is from paternal allele, one is from maternal allele. This person also has gonads, right, or the germ plasm. In the germ plasm, the person will make gametes, say sperm. Another individual, same germ plasm, a female, assume that, and that will make egg. And fertilization would occur, a zygote would form. The zygote will undergo stages like morula, blastula, neurulation, and so on. During those times, there are two things that will happen. You will have primordial germ layers, which will be, say, ectoderm, mesoderm, endoderm. And this will form the somatoplasm, all the body that we have. And there is also one more thing that will form is primordial germ cells, from which we get the gametes, right? This is going to form the germ plasm of that individual. Primordial germ layers, primordial germ cells. Primordial germ layers will give rise to somatoplasm. Primordial germ cells will give rise to the germ plasm. So what we are discussing right now is about the cycle of genomic imprinting. When does this imprint made? I'm not going to discuss the importance of it and other things, but because we want to discuss about how to study genomic imprinting, and that is a good part of thinking well. So what happens during these primordial germ layers is, when an individual is forming, or the germ plasm in the beginning stages when it is forming, what happens is the germ plasm is coming from primordial germ cells, and that will give rise to gametes. For simplicity, what I'll do is I will write the sperm, and sperm will fuse and form a zygote that is 2n, which will form the somatoplasm and the primordial germ cells. Okay, this is the cycle that happens, right? We are all, each generation after generation, this is what happens. Primordial germ cells that is present in an individual will give rise to sperm. Sperm will give rise to a zygote. This is the body. This is the individual in which new primordial germ cells are forming, and they will form next generation, right? So with regard to genomic imprinting, okay, during this stage, the we are saying imprinting as status of DNA methylation. There are other mechanisms, but we are just sticking to DNA methylation alone. During the formation of primordial germ cells, all imprints are erased. Remember that. It is called as erasure. Okay, imprint erasure. And this step is gametogenesis. From primordial germ cells, we get the gametes, and gametogenesis we will call it. If it is sperm forming sperm, we will call it as spermatogenesis. If it is egg, we would have called it as oogenesis. During gametogenesis, the imprints are established. Okay, so there are two things. We'll look at what would happen if it were DNA. So we'll take it as the genes A and B. Okay. We will start here. Say I'll just write A and B. In A and B, what is the status of imprint? We said it is erasure. That means, what can you say? Say in the let me go here. In this state, I told that in this case A is methylated, and in the maternal allele B is methylated. Now let's go with the same thing with A and B. I'm saying it is erasure. So which of these genes are methylated? A and B in say maternal allele, paternal allele. Which of these genes are methylated? Can anybody say?
Neither of them, sir.
That's right. Erasure, I said, it is all the methylation is removed. So A, B, A, B, none of them are methylated. In this sperm, we would have, they are haploid, isn't it? So we would have A and B. And we are saying that the imprint is established. So what would you expect? Which one of it is methylated? You can also take a clue from here. When we are saying it is a
A.
sperm, it is male, that is right. A is methylated and B will remain unmethylated. And somewhere, say from another individual, we will get the egg. In the egg, what would you expect? It is also haploid, A and B. Which one is methylated? Which one is unmethylated?
B should be methylated.
Yes, because B is maternally imprinted, egg is coming from the female, so B is methylated and A is in unmethylated state. And now we have a zygote. In zygote, it is 2N diploid, so we will have A and B. One is coming from the maternal allele, another allele or another set is coming from the paternal allele. So which ones are imprinted here? Based on this one, we'll say B is imprinted here. And from the sperm, A is imprinted. Okay? And the whole body that is developing will have the same, the somatoplasm that we have in our body is made from the gametes of our parents. Whatever signatures they have put in, that is what our body is made up of. In our primordial germ cells, we are making in the new imprint. Whatever our parents have put in the imprints, we erase them all during our early stages, or when we are forming from the primordial germ cells. We erase all the imprints that we inherited from the parents, and we make our new, and that is what we are. This is something that is made of us. The body that we have in us, excuse me, sorry. So the body that we have, the imprints are made by our parents during gametogenesis. Okay, it's a nice way to look at it. It's a complex phenomenon. Why it happens? There are many theories you can go around. I hope you understood this set. What we will do is a complex problem, and I will give you a quick guide, or maybe we'll try to solve a question. Usually, did we solve this question before or not? Anybody? I don't think we did. Maybe I should have done it a long time ago. But I will go ahead. I will also try to maybe help you recollect about restriction endonucleases. Right? We discussed about restriction enzymes. While discussing about restriction enzymes, I mean, there are endonucleases. That is one thing. and they are sequence specific. We also discussed their palindromic sequences, 4 base cutters, 6 base cutters, 8 base cutters, and many other things. While discussing that, we also discussed something else with regard to methylation. Methylation, based on methylation of the target, we discussed something. I hope you can recollect that. The first thing is methylation sensitive, then methylation insensitive, and then methylation dependent. Right? What does that mean? We will have a quick discussion on that. When we say methylation sensitive, so if there is a DNA that is present and the DNA is the restriction enzymes with our methylation sensitive can cleave DNA that are unmethylated. I will do something like this. Unmethylated targets, and I will write as methylated targets. Sensitive ones can cleave only unmethylated target sequences. They cannot cleave methylated target sequences. That's why we write it as, we call them methylation sensitive. Methylation insensitive, that means they will cleave whether the target sequence is methylated or unmethylated. It doesn't matter. They would just cleave them, the target sequences. Whereas restriction endonucleases that are methylation dependent cannot cleave unmethylated DNA. They will only cleave if the target sequence is methylated. Is that clear? I hope so. So we can use different restriction endonucleases and based on their sensitivities as well. Okay, sensitive, insensitive, and dependent. I'm not going to discuss about specific examples of restriction endonucleases and other things. But what we will want to is be able to solve problems, the capacity to solve that. So we will do a small exercise and let's see how we go ahead with that. Sperm, I will say egg, I will say somatic cells, and then primordial germ cells. Okay, for ease, well, let's try solving it. There is also one more thing I would want you to remember. Yeah, sorry. We also have one more aspect to write here. We are cutting we would have isolated DNA from each of them, and then we would have okay. So what we are going to do is we will take this one second. Something is not working. We would have isolated the DNA from them, and then we would have the procedure for southern blotting, you remember, right? We will take the genomic DNA, we would have digested with restriction endonucleases, then electrophoresis, and then we would have done southern blotting. In blotting, we would have used a probe, right? The probe is, I will give you the conditions that you can understand, and then we will see how we will go about it. Okay, something is playing up too much. Let me try this. I will give you all the details and we will work through the problem. and it should be a lot of fun if you participate along with me. Okay, the experimental setup is southern blotting, genomic DNA, restriction endonucleases. In that, we will use 3 different endonucleases. These sensitive, I will call it as P, or I'll call it as S. Okay, I can also use insensitive one. I will call it as I. I can also use methylation dependent one. I'll call it as D. So I hope you understand SID, what it means: sensitive, insensitive, dependent in all the cases. That is one part of the question that you need to remember. I will also draw the genetic architecture like we have already discussed. There are 2 alleles. One is from maternal allele and another one is from paternal allele. But I will just, for the description, I will just give it as gene A and gene B. And here are the restriction sites. I'll use a different color. This is a restriction site, and this one is another restriction site. Both, for the sake of our experiment, I'm going to use, so the S, I, and D, all these 3 restriction endonucleases recognize the same sequence. But then how are they different? One of it is sensitive to methylation, insensitive to methylation, and dependent on methylation. So they are different with regard to methylation status of the target. Okay. Yes, they recognize the same sequence, but they are different in the way they behave based on the target sequence methylated. So this is 2 kb, this is 3 kb, and this one is 4 kb. If we cut using any of these enzymes, typically, we should get, say, something like 2, 4, and 3. Okay? And I need to also give you a probe. Probe, assume that it is binding to A. Okay? I hope you remember the southern blotting experiments we have done. Only thing we are doing it different here is you need to keep track of the sample. We need to keep track of where the genomic DNA, whether genomic imprinting is established or not. We also have to keep track of the which enzymes we are using, what is the nature of them. And the second one, of course, is we have to remember which one is haploid and which one is diploid. This is haploid. This one, egg, is haploid. Somatic cells, diploid. Primordial germ cells, diploid. Okay. So if I have taken sperm and I'm giving you already the data we said in the previous experiment, what we discussed is A is methylated on the paternal allele and B is methylated on the maternal allele. Okay. So that same information you should say A paternally methylated or imprinted, I can call it. B is maternally imprinted. Okay. Now I will just try to solve with the sperm. If I have collected the sperm and isolated the genomic DNA and try to run the gel, okay, perform the southern blotting using probe P, okay, what would you expect in case of S? Or for simplicity, I will ask, what would you expect if I used insensitive, methylation insensitive restriction endonuclease? If I cut it, where will it cut? Can you tell anybody? What does I say, insensitive say? It means it will cut whether the
We'll have all
DNA,
the 3.
yes, whether the DNA is methylated or not, it would cut. It would cut here and it will also cut here. So what will be the fragments that will be visible after performing southern blotting?
Only one fragment, size 4.
Yes, size is, yeah, you will get only one fragment, that is 4. Why is that? Because cut has been made. There are 3 fragments, 2, 4, and 3. But on probe is binding only to this fragment where gene A is present. Therefore, you will only see that fragment. Right? Good. So if I cut the DNA using sensitive one, what would you expect? What will be the size of the band?
7, sir.
Why is that?
Because it would not be able to cleave the cleave at the second site, second restriction site, because B is getting methylated over there.
Why is B methylated?
Oh, sorry, sir. It's A is getting methylated
Why is
because
A
it's
getting methylated?
because it's paternally imprinted.
So,
So it should be
yes, you're
6 and not 7.
you're right. Because the sample is coming from sperm, which is coming from the male. I understand you're thinking, you might be thinking where one is sperm and egg, how can one individual have? Assume there is a hermaphrodite, and that is like earthworm, for example, which can have gonads for both the sexes. It's a theoretical problem. You will see the complex one in the next question. A similar one, basically. It's a practice one. So in sensitive ones, because it is coming from a sperm and it says A is imprinted on the paternal allele, so you can expect that there will be methylation on A. And B is maternally imprinted, so should be no methylation here. Therefore, what you can expect is the sensitive enzyme would cleave here. And so we will get a fragment that is about 6 kb. Okay. What would happen if we have digested it with D? What fragment would you expect? D is, it is dependent on methylation.
We'll have a 7 kb fragment.
That's right. We will get a 7 kb fragment here. Okay. So can you tell, can others also please try for egg? What would we expect? In the egg, the methylation pattern will be different, isn't it? In the egg, the methylation pattern would be, because egg is coming from a female, A would be unmethylated state, whereas B would be in methylated state. So if we digest it with I, where it's insensitive, we will get the same fragments, like it will cleave on both the cases here as well as here. So we will get a 4 kb fragment, similar to what we got for the sperm. If you have digested with the sensitive restriction enzyme, methylation sensitive restriction endonuclease, then it will cleave here near A, but not here. So we will get a 7 kb fragment. And if we use the methylation dependent one, because it will cleave only here because there is methylation, so we will get a 6 kb fragment, right? So now we have to do with somatic cells. Somatic cells is a little bit complex. Not complex, actually. They are biparental. And the important thing is that they are diploid biparental and they have imprints that have been inherited from the parents. So there are 2 alleles. One is from the paternal allele, the other one from the maternal allele, gene A, gene B, gene A, and gene B. From this sperm, we would have gotten a an imprinted one like this. So this is 2, this would be 4 and 3. From the maternal allele, we would have gotten this region methylated, and this one is free. 2, 4, and 3. Now, if we digested this DNA, diploid DNA, with insensitive one, what are the bands that we expect?
4 kb band.
Yes. Insensitive one, we will get 4 kb fragment only, because insensitive enzyme would cleave here or and here, here and here. So the probe is binding only to this fragment. So we will be able to visualize 4 kb fragment. With methylation sensitive enzyme, what would you get? What fragments would we expect?
6 and 7 kb fragments.
Yes, methylation sensitive one will cleave here for the paternal set, and it will cleave here. It will cleave only the unmethylated regions. Now we have 4 plus 2, 6 kb fragment, 4 plus 3, and 7 kb fragment. So we should be able to expect 6 and 7 kb fragment. Okay. And if it is methylation dependent one, what would we expect? It would cleave all the methylated regions here and here. Then also we would get, similar to that band, we'll get a 6 kb fragment and a 7 kb fragment. What would happen in primordial germ cell stage? In primordial germ cell stage, what is the thing we discussed about the imprinting status? There is erasure. All the imprints that we have inherited from our parents are erased, and we will make new imprints based on the sex of our own sex, either male or female. So if this is it is diploid in state, but there is no methylation anywhere, because because there is erasure of all the imprints. So now if we treat it with I, what would we get? What band will we see?
4 kb fragment.
We will get a 4 kb fragment. With sensitive one, methylation sensitive,
Again a 4 kb fragment.
that's right. And methylation dependent,
No fragment at all.
yes.
We would not get any fragments, sir.
No, assume this is the genome, the total
Oh,
length.
the overall 9. Yes.
Yeah, we will get a 9 kb fragment, right? And that is how we can study things. But the point is, how many things do we have to keep in mind for this? The first important thing is that, I mean, there are so many things, and it's like a puzzle or a game that we have to solve, and we need to keep the status of the sample type, as I was describing. Is it haploid or diploid? Is it egg or sperm? Is it at the stage of, is it a somatic cell, or is it primordial germ cells? Right? That is what we have to know about all the sample. About the restriction endonucleases, we should know about sensitive, methylation sensitive, methylation insensitive, and methylation dependency. And about the genes, we should know which of, whether they are paternally imprinted or maternally imprinted. And we should also know the fragment sizes, right? What are the fragment sizes? And we should know about the probe. Where does it probe actually bind to? Imagine if you are able to practice more questions on this, and it is just so many things, and you're trying to decipher the correct answer. So I will give you a question that is made, but you have all the information the same as what we have already discussed. There could be a few changes, except that this there is a small error in this. It's written at 11, but it's actually 15. Okay, 15 kb fragment is given. Here the question is different. We said about gene P and gene Q. The sites are given as dotted lines. The sizes are given here. And there are three restriction endonucleases, X, Y, and Z. X is, I think the details are here. X is methylation dependent, Y is methylation insensitive, and Z is methylation sensitive. And the probe is binding to gene P. So gene P and gene Q, they are imprinted genes. So one is methylated paternally and the other is methylated maternally. But we are not telling you which one is maternally imprinted or which one is paternally imprinted. All you are given is gene P and Q, out of which one is maternally imprinted and one is paternally imprinted. That is given to you. We, based on the information that is given here, and one band is given to you here as a clue, from which you have to work out the whole banding pattern in all the cases here. Okay, can you do that? It's all the process slow. We have gone through a process and I have been using a lot of rough work. If you can do like that and use as many papers as you can, you are likely to get the answer correct. Please try to do this. It's a very interesting, and I think it will improve your competence in thinking a lot. And this is one of the techniques used in studying imprinting or epigenetic mechanisms also, where we have to study about DNA methylation. The other way of studying methylation status is bisulfite sequencing. Please have a look at it, and we will discuss. We will not discuss, I will directly ask in the exam. You should know the principle of bisulfite sequencing. Using restriction endonucleases, using southern blot, how can we do, how can we read about the methylation status, DNA methylation status of a particular area? If you can do this one, you would have understood a lot of things, and it's a fantastic capacity to think multiple factors and arrive at the answer. I'll stop here. If you have any questions, I'll be happy to answer. But otherwise, I want you to start this answer this question. I'll also post this on the Google Classroom. Please try to upload there. And I thank those who have already uploaded some of the assignments from the previous one. That's very good job. Thank you very much for uploading your answers. Yes, any questions? If not, this is the end of the class, and I think we will also have some face-to-face classes. We can discuss more questions. I would also encourage you to come up with any CSAR questions which are between molecular biology and genetic engineering, especially from part C. If you can come up with any of those questions, we can always solve and have fun. Okay. Yes, thank you very much for attending then. Yeah, thank you. Yeah, watch the videos. I'll post other ones as well. Thanks.